Hello, Evelyn. Can you hear me?
Yes, sir.
All right. I'm going to go over for everyone that's watching, I'm going to go over what we have in store for this week. So please keep in mind that this week, the week of the 6th, we have exam number 2 due. There's also 2 opportunities for extra credit, and it's going to be the neurophysiology and somatosensory pathways. But I guess the gist of it is that exam number 2 is going to be due at the end of the week. So keep that in mind and adequate time accordingly. For lab, we will this is what we're going to do at the end of the week, but there is no quizzes due. Our next lab practical will be due next week. All right, so what are we going to do this week? This week we're going to cover chapter 14 as well as chapter 15, and it's going to be all nervous system. So without, there's a lot to cover, so I'm going to just get on with it. So here we go. We're gonna cover the brain and cranial nerves. There's a lot of structures that you're gonna have to become familiar with. Some of the names might sound familiar. You probably heard of hippocampus, thalamus, hypothalamus. Those should ring a bell. But for the most part, it's challenging to just identify where they are and what they do because of not only the histological homogeneity of the brain, but also the overlapping functions that a lot of these structures have. You will have your hippocampus be involved in memory, your hippocampus be involved in motivation, involved in emotions, involved in injury responses through cell division. All of those things, they're not clearly and easily classifiable. Same goes for regions of your brain cortex. Usually everything that you do, everything that you consider yourself is the accumulation of experience and the constant interplay of different regions of your brain, different neurons firing at the same time. Myself sitting in a chair, being aware of my environment involves like my visual system, being familiar, bringing this visual information originally, cementing it in some sort of memory, categorizing it as a room. That way, every time I go into a room, I don't have to make up a decision. What is this? Is it a room or is it a dog? Your brain does all of that, does all of that subconsciously. And you don't have to think about all of those decisions, all of those categorizations that happen. That's great because it allows us to not dwell in minutiae as we enter every single environment, but it's not great as well because we tend to be over-categorizers. We tend to, everything usually, the notion of stereotypes comes from this natural propensity for us to just have a categorization for everything. We enjoy also having answers. We are not too comfortable with the unknown. That's also why a lot of times throughout history we've been providing answers to things that we don't know, even though the best answer could be we don't know. But for the most part, we do assign a response is just part of human nature. But overall, I hope part of that you took that your brain is all of the, it has many structures that are involving many functions, and all of those functions are what allow you to perform these really complex tasks in almost in a seamless manner. Your visual auditory system, sensory, somatic, your sensory system, all are playing a role right now when you're listening to my voice, and it seems like a seamless act, the act of listening. If you do it right, you will engage portions of your motivational limbic system that will allow you to, that provides that more weight into this moment, into these words, as opposed to the way that we provide to, I don't know, a binge, whenever we're binge watching and something on Netflix, and we're just like, it's in the background, we're kind of paying attention, we listen, we hear, but it doesn't, it's not recorded into our memory bank. In order for us to record that into our memory bank, our limbic system has to have the motivation to do it. It has to understand the weight of it, and that recruits more resources into storing this information. That is why we humans, most of us, for example, if I ask you, what did you have for lunch 2 days ago? You really have to dig into your memory banks for that. If I ask you a week ago, that's a nearly impossible task. A week from today, what did you have for lunch? Unless there was a major event that happened there, you won't remember, and it is because we allocate resources based on the allocate resources based on our brain's computing power. We simply cannot allocate resources to having all of this biographical information be stored within our brains without it interfering with other functions. If we were to have this type of just endless biographical information stored in our brains, every time we are involved in the present, we're involved in a new conversation, or just a regular conversation, then if we were to store everything everything that has gone through the duration of our lives, and each single word we cannot trigger a memory. And usually we do that whenever we're in a conversation. You know, you talked about something and then something triggers you, and you provide your input towards that conversation. But those are usually meaningful events in your life that you remember. But what if you remember everything? And there were like 20 memories that were triggered, or maybe hundreds of memories that were triggered as you are within a conversation. Now you have to decide, okay, which one of these was relevant and which wasn't. I have 100 memories in my head that I could input here, and that would be you would engage a lot of energy in order to undergo those decisions. So anyway, being forgetful, it's a blessing and a curse, because you want to remember things whenever you are studying, whenever you're reading something, but we cannot do it on cue. You have to really practice this art of focusing. Whenever we talk about focusing, it's merely your limbic system, these motivational aspects of your brain telling you, hey, this is important. We need to pay attention to this. We need, your resources are allocated towards that. You cannot block out inputs. That's how important what you're engaged in is. And I guess you kind of, you understand that the fact that we're blocking inputs kind of hints at you that that you are focused on the task, but it is us basically that that elusive focus on motivation. That's how we experience it. But anyway, that being said, I hope out of that you got that we're complex and that it's hard to study. Okay, but you're gonna come. There's a lot of new terms whenever we're going through these chapters, and hopefully you're able to focus. Or otherwise, just try to repeat and try to go engage with the material as many times as you can and in as many different ways as you can. Okay. So let's go. Let's start with the brain. We have 6 major regions. You have your cerebrum, which is what most of us understand as brain. 2 hemispheres with longitude separated by this longitudinal fissure. Have a diencephalon containing the thalamus, hypothalamus, and epithalamus, or your pineal gland. You have your mesencephalon, which is your midbrain. You have the pons. You have your cerebellum and your medulla oblongata. The medulla oblongata, the pons, and are going to be part of the what's called the brainstem. And your cerebellum, just like your cerebrum, is going to be divided in hemispheres, to 2 separate hemispheres. All right, so a lot of new terms of the get-go: medulla oblongata, pons, mesencephalon, diencephalon. Those are the big structures. We're going to dive into these big structures and understand the components that make up these big structures. Whenever possible, we're going to go back into the function. That way, it can hopefully that would help cement this knowledge. Okay, so the brain contains a fair amount of neural cortex. You have the distinction between, you have gray matter on the exterior portion of the brain. In this gray matter defined as cell bodies of neurons, and white matter defined as the axons that connect these neurons. So in this neural cortex, you would have, there's usually, the neural cortex has 6 clearly defined layers of neurons. And those 6 clearly defined layers of neurons communicate with other regions of the brain, within the brain cortex itself, within the diencephalon, within other areas, in order to perform their function. But here's just a view of the anatomy of the brain. These are we went over this during lab, and I hope you become familiar with these terms. Here I'm gonna point out the medulla oblongata on the pons, part of the brainstem, and the cerebellum. Here, the cerebellum has been removed to get. Oh, sorry. In this view, this is an anterior view, so you cannot see only a small portion of the cerebellum from this view. But from the posterior view, you get to see the cerebellum and all of its rugae and folds. Most of the neurons in your brain are going to be within the cerebellum. It's a fairly dense part of your brain. So here we have, you have this gyra and sulci. You have The elevations are going to be called gyra. The indentations are going to be called sulci. If you have deep indentations, those are going to be known as fissures. There's three main fissures that divide the left and right hemisphere of the brain, the left and right cerebellum, and the cerebellum from the cerebrum itself. This layer that you observe here is going to be part of the meninges. The meningeal layer that is in direct contact with the brain is known as the pia mater, and this is what we are observing at the moment. All right, now we're going to look at different specialized functional layer regions of the brain. So you have here your you have your central sulcus present here. You have your have a central sulcus, you have a lateral sulcus here, and you have a parieto-occipital sulcus. Those sulcus are there to separate. They help you identify these different lobes in the brain. So central sulcus will separate the frontal and parietal lobes. Your lateral sulcus will separate the temporal from frontal lobe. And your parieto-occipital will separate the occipital from the parietal lobe. Here you have your different lobes. In this view, you see the medulla oblongata pons, and you see the midbrain as a deeper structure, but that is the brain stem. So it kind of makes sense. It's the stem of your brain, so and the medulla oblongata is continuous with the spinal cord. So one thing that I want you to appreciate here is you have here your medulla oblongata, the spinal cord. The spinal cord is going to be sending signals from basically everywhere in your body, right? And it's going to send signals, sensory signals, sensory inputs, and a lot of them are going to synapse with neurons either in the medulla oblongata and the pons. Now, these sensory signals are going to be sent to the cerebrum. You see how closely opposed they are. We will see later that the pons has these thick bundles of axons called peduncles. And that is because there's a lot of communication occurring between the cerebellum and these regions of the brainstem. The reason being is because the cerebellum is going to be involved in motor coordination. In order for motor coordination to occur, you're going to have sensory inputs being sent to the cerebellum and motor outputs being sent the other way. And whenever there is all of these sensory inputs and outputs are going to come and go through a set of axons called the peduncles. We will understand later why how the cerebellum performs its role. All right, these are the 6 major areas that we described. So here's the cerebrum with all of its different specialized lobes and regions and specialized regions within the cortex. Here's the cerebellum. Coordinates complex somatic motor patterns. We'll look at that whenever we reach cerebellum. But the cerebrum is all of these sensory signals that you have, all of the sensory inputs that you receive are going to eventually kind of synapse in the cortex of your cerebrum, and from there is going to associate with other areas. So that way, the auditory system doesn't exist independently of that way you can associate all of these inputs and provide richer context of audio, visual, respiratory, and so forth. Once you get this input, the sensory input into your brain, some you will store. You will go ahead and store it in a short-term memory that if it gets exercised enough, it will eventually go into long-term memory storage. So this is, and also here all sensory input and output from the muscle system. The cerebellum, we mentioned, coordinates complex movement, and it helps you adjust. So you have motor outputs as being sent from the prefrontal gyrus, which is known as the primary motor cortex present here. You have here your central sulci, your central sulcus, and in front of it is the motor coordination of your brain. So this is the primary motor cortex. The primary motor cortex is the reason why I raise my hand right now. The reason why I raise my hand so smoothly and without trembling is because, or in such a coordinated manner, is because of the cerebellum. Cerebellum, it's kind of like it's constantly storing movement, motor information, sensory motor information. And whenever you initiate a movement, the cerebellum provides the input because it retains information about how to perform that movement in a coordinated manner. The notion of muscle memory is the cerebellum at work. A lot of us are left-handed. If I were to try to pitch a ball with my left hand, it's not a gracious movement. If I pitch a ball with my right hand, I'll probably get closer to the goal. Now, why is that? I know consciously how to perform that movement. I know that it goes there, and I have my visual system working in the same way, whether I perform that throw with my right over my left hand, yet a throw with the left hand is not as coordinated. That is because every time I throw with my right hand, I bet the first throw wasn't coordinated either. But every time I repeat this motion, basically my brain is coordinating the movement. My primary motor cortex is saying, make this coordinated movement and throw the ball in that direction. My visual system is providing feedback saying, okay, that went well, that didn't. But all throughout this movement that I'm repeating, the cerebellum is storing information. The cerebellum is storing the information that whenever my bicep retracted, I had relaxation of antagonistic muscles, and it stores all of that information. So as soon as you repeated that, the information, that blueprint of that movement becomes more coordinated. The neurons that fire whenever we perform the movement right begin firing almost exclusively without the noise from other neurons surrounding it. And that is what the cerebellum does. So your primary motor cortex initiates the movement. However, the cerebellum is the one that coordinates it. As the movement gets sent to your muscles, the cerebellum has input into how that movement is going to play out. And here is the cerebellum, and it sends all of those. The neurons present there send axons through these peduncles present here. So all of that communication that occurs between the cerebellum and the pons or the medulla, it's done through these. Here they are. These peduncles, these thick bundles of axons. But hopefully that kind of helps you understand the role of the cerebellum and kind of why we have these thick peduncles there. All right. The medulla oblongata relays sensory information to thalamus and other portions of the brainstem. So you have sensory information coming from all your regions of your body, makes a pit stop at the medulla oblongata, because there are several, in order for the sensation in the tip of my finger to arise to its almost nearly final destination in the post central gyrus, or the primary sensory cortex present behind the central gyrus, in order for that electrical signal that initiated here, that action potential that initiated in the tip of my finger, to reach that level, it has to go through several synapses. It's primary neurons synapsing with secondary, tertiary, quaternary, and quaternary neurons, in order for the signal to finally reach destinations here. And then from, once it reaches the primary sensory cortex, it will go to association areas present here in the parietal lobe. So there's like fifth order neurons, if you can think of it. All right, so let's go. The medulla oblongata is part of that relay system. So sensory neurons will make a synapse. Sensory neurons that exist in ganglia outside of the central nervous system will send a signal to the spinal cord, from the spinal cord to the medulla oblongata, from the medulla oblongata to the thalamus. One theme that we will hear regarding sensory information is that it usually makes a pit stop at the thalamus. The thalamus is the place where this sensory information gets imbued with a context of emotion. What is emotions? And that is the reason why whenever you smell something sweet, a rose, it might bring some fond memory, or whenever you smell something disgusting, it might bring a national, just emotional level of aversion. But once again, what are emotions? What are these? I mean, we understand them probably more in context of the definition of the word, and not in but outside of the biological context of it. Within the biological context of emotions, emotions are these strong drivers of behavior. For example, love is a very strong emotion, and because of love, we do a lot of things that are not rational. There are other emotions that promote not rational behavior. However, whenever you think about what's not rational, it's something that overrides critical thought. The way that a good way to understand emotions is shortcuts. Shortcuts within your brain that are primed by that arise from primal evolutionary behaviors. Primal evolutionary behaviors such as the behavior reproduction. Reproduction is a primal evolutionary behavior that is, whenever it comes to, yeah, I was gonna run, but we'll leave that for another time. Let's so anyway, that's the thalamus. The thalamus and the limbic system are playing a role within emotional control. Whenever you think about emotion, this is our evolutionary, more ancient structures within our brain, what's commonly known as the reptilian system. These are basically, what they mean by that is that we share these common structures with with ancestors that we share these structures with our more ancient ancestors, if you will. And these structures provided us a selective advantage. That is why we are here, and that's why we're not dodos or other extinct species. And those selective advantages, we pay attention to them. We take them seriously. They override some of these more critical behaviors. All right, let's move on. So here we have the pons. It also relays sensory information, and once it does, sends it to the cerebellum and the thalamus. It sends it to the cerebellum for motor coordination. It sends it to the thalamus once again, because this is how our reptilian ancestors did it, and it worked out well for them. We kept the formula. We still do it. Downside of it is that sometimes that reptilian brain takes over our rational thinking, but we're still alive, which is they have more benefits than downsides, I reckon. Mesencephalon, here you have, this is your midbrain. This is part of your brainstem, your medulla oblongata, your pons, and your mesencephalon present here. Your mesencephalon is going to have a function within the processing of visual and auditory data through the superior and inferior colliguli. You have some reflexive responses, and it is involved in the maintenance of consciousness. It's that it's kind of like the starter of your car. That's the mesencephalon for us. It's the starter of our consciousness. Gotta crank it up. So here is the diencephalon. The diencephalon, it's all of these structures are embryologically more ancient than your cerebrum itself. Here you have the diencephalon. So you can think of it. Whenever we build our nervous system, we build structures on top of older structures. Here you have the thalamus, is the relay, and it relays and senses, is the relay for sensory information. Once again, these were sensory, or this information is imbued with sense emotional context. And the hypothalamus is involved in, it's also involved in this emotional behaviors, and it helps control autonomic functions, and this is the initiator of hormonal system. You know, your pituitary is the master regulatory gland of your body, and it's because it produces several hormones that are involved in maintenance of several homeostatic processes. But the hypothalamus is the structures that controls the master regulatory gland. So it's the master of the master regulatory gland. Is that it does so by basically telling the pituitary to either release or not release certain hormones. Okay, those are the main structures of the brain. Now let's look at a little closer to their roles. During embryological development, we develop from a tube, basically, called a neural tube. This tube becomes more complex from anterior to posterior, this tube is divided into the prosencephalon, mesencephalon, and the rhombencephalon. The prosencephalon is going to give rise to your cerebrum and your diencephalon, your brain cortex, your cerebrum, and your thalamus and hypothalamus. Your mesencephalon is going to give rise to the midbrain, or the and your rhomboencephalon is going to give rise to your medulla oblongata and cerebellum, and pons, cerebellum, pons, and medulla oblongata. It gets divided into the metencephalon and myencephalon. Just quick, something to help you remember is that the mesencephalon will give rise to the mesencephalon. There's no division here. And the rhomboencephalon, or most posterior portion, it makes sense that it gives rise to the most posterior portions, if you will, being the cerebellum, the medulla oblongata, and the pons, right? The medulla oblongata comes from the myencephalon, and the cerebellum and pons from the metencephalon. Let's look at the ventricle. So as this tube kind of develops, the tube has a lumen in the middle of it, and this lumen persists throughout development, and it remains with us as adults. This lumen is going to give rise to the ventricles of the brain. So let's look at the ventricles of the brain. So remember that we were once a tube, right? Just a long tube. Here you have this tube became this really complex cavity with that contains cerebrospinal fluid. But the portions of the tube that were associated with for example, the prosencephalon, the portions of the tube of the lumen that were associated with the prosencephalon remain associated with the rhomboencephalon throughout as we develop more into more complex structures. So here the lateral ventricles and the third ventricle, both of those are still associated with the cerebrum and the diencephalon. You also have here the so just by you can infer by that the rhomboencephalon, the lumen of it was the lumen that became the lateral ventricles and the third ventricle. So here you have the fourth ventricle is between the medulla and the pons, the cerebellum, pons, and medulla, and that's going to be the lumen that was shared by the rhomboencephalon, and the midbrain is going to be associated with the cerebral aqueduct, and that is the lumen that was shared by the mesencephalon. So I hope that you realize that we were just a tube with like a chewy center, and then that tube became wrapped around and it became complex, but the inner portion of the tube that contained the cerebrospinal fluid gave rise to that complex series of cavities that contains cerebrospinal fluid. We went through this. I'm going to touch over briefly. These are the meninges. You're going to have a dura mater, an arachnoid, and a pia mater. The dura mater is going to contain two layers, one of them that's in contact with the bone, a periostal, and one of them that's in contact with the other, the arachnoid in this case, the other meninges, and that's the meningeal layer. Between these two layers of dura mater, there's the dural space. This dural space is, this dural sinus contains venous circulation. Basically, drains the brain of its circulation and then takes it through, takes it back into the heart. The arachnoid contains between, so here's the arachnoid. It contains what's called a subarachnoid space between the arachnoid and the pia mater. So subarachnoid, below the arachnoid, makes sense. Subarachnoid space, and this space is filled with cerebrospinal fluid. Now, here we have the pia mater is the meninges that's in direct contact with the brain. Astrocytes help adhere that pia mater. They basically hold on to the pia mater so that you have your cerebrum and your pia mater bonding to each other. Here you have these infoldings of the meninges. These are the features that we talked about. They are kind of filled with this infolding of the meninges. So basically, this infolding of the meninges is in the longitudinal fissure, which separates the left and right hemisphere. This infolding present here, and this is known as the falx cerebri. You have the tentorium cerebelli present in the parieto-occipital fissure. And you have the falx cerebelli. This is for the cerebella that is present in the cerebelli fissure that divides it into left and right hemisphere. This is a view of the brainstem. You have here the spinal cord, the medulla oblongata, the pons, and here you have the midbrain present here. This is a view of the diencephalon. So this is the midbrain. The brainstem is only going to be the midbrain, pons, and medulla oblongata. Okay, those three components. This is a posterior view that allows us to see all of the axons. These peduncles that you observe here, those are all of the axons that are going to and from the cerebellum. We mentioned that cerebellum has an input in motor coordination. In order for it to have an input in motor coordination, it needs to know, it needs to have sensory input to know, okay, whenever I throw this, my arm is going this way and it extended fully. So it needs that sensory input, but also it modulates the sensory output for coordination. All of these structures we mentioned that they share a lumen, and here is you don't see the lumen here, but what they're trying to illustrate here is that the lumen of the ventricles and central canal present in the central nervous system contain this specialized. First of all, it's completely surrounded by ependymal cells that help in maintain this barrier between the cerebrospinal fluid and the brain. And there is a specialized cell populations of ependymal cells called the choroid plexus. This choroid plexus is depicted in red because it's highly vascularized. In order, we mentioned, the choroid plexus creates the cerebrospinal fluid. What's the cerebrospinal fluid? It's liquid, water. Where does water come from? We get water from the circulation, from the plasma. So it has to be richly it has to be richly vascularized in order for this process to occur, in order for the cell, in order for cerebrospinal fluid to form. And that's what's being depicted here. At the diencephalon facing the third ventricle, we have choroid plexus making cerebrospinal fluid. Here in the fourth ventricle, you also have choroid plexus making cerebrospinal fluid. What does the cerebrospinal fluid do? cushions support the brain and transport substances. There's been known to be neurotransmitters within cerebrospinal fluid. They perform, so it performs these functions not only to help modulate and the environment within the brain. All right, so we mentioned that cerebrospinal fluid is formed here by the choroid plexus, and it's released into the ventricles, into this empty space. So here's the cerebrospinal fluid flowing, and within the ventricles and the central canal of the spinal cord. Now, how does it get from the ventricles and central canal to the subarachnoid space? How does it get from the interior of the brain, which is these lumens, this cavity system in the inside of the brain, to the meninges, which are on the outside of the brain? It does so through these apertures within the fourth ventricle here. Here you have in the fourth ventricle and the cerebral aqueduct. Here you have the lateral aperture present here and the median aperture present here. These apertures are contiguous with the subarachnoid space, and cerebrospinal fluid is flowing from there to the subarachnoid space. Eventually, from the subarachnoid space, here it is, the subarachnoid space is going to, we mentioned that there is between the two layers of dura mater, there is a space which contains venous circulation. That is where there is this specialized structures called arachnoid granulations. And those arachnoid granulations are where the cerebrospinal fluid returns to the circulation. All right, let me jog a note through this. Diencephalon and brainstem. We mentioned all of these components. You see here all of several of the cranial nerves are going to synapse within the pons, the brainstem, and the midbrain. So here you have them. So these sensory inputs and outputs are going to be synapsing in our brainstem, in this older structure, older evolutionary structure. Here you have a view of the relationship between, this lateral view probably gives you some of the best appreciation of how they relate to each other. The medulla oblongata, the pons, and with all of these peduncles or bundles of axons coming and going from the pons to the cerebellum. And here your midbrain, containing also peduncles, this thick mass of axons going into the brain itself. All right, let's look at some of the functions of the brainstem. The pons. This is a view of the medulla oblongata. The medulla oblongata helps us as a relay center for sensory input, reflexes, and it contains a cardiovascular and respiratory rhythm centers, as well as centers for digestive activity. So here are your cardiovascular centers depicted in red, and your respiratory rhythm centers depicted in orange. So you don't have to think about breathing 14 times in a minute because of these autonomic functions provided by your medulla oblongata. The pons, we mentioned that here you have the four cranial nerves entering the pons. It also helps with the control of respiration and the tracks that are there linking this normal process between the sensory input and output that occurs, at the source and your primary motor cortex, and your primary sensory cortex. The cerebellum plays a role within this, and that is it links to this process through the pons. Here's the cerebellum, responsible for voluntary movement coordination. Please be familiar with this anatomical features of the cerebellum. Just like your cerebrum, it does have its own cortex, its own medulla. It has several lobes, and it has this. The cortex itself is going to contain just a thick, dense population of neurons, and this arbor vitae, which is going to be the white matter or axons that transport the information. Diencephalon, epithalamus, hypothalamus, thalamus. We mentioned that the hypothalamus highly involved in the endocrine and nervous system. Hypothalamus, the regulator or the master regulatory glands, secrete hormones, coordinates autonomic movement, involved in body regulation. Thalamus acts as a relay center for sensory information. We mentioned sensory information makes a pit stop at the thalamus. Thalamus is involved in the limbic system, this system for motivation and emotion. We're going to talk about it later, but it makes sense that in order for us to perform any sort of function, it involves motivation that we are aware of. Usually the trigger for those functions come from our senses. They rarely come from some innate place within your brain. There's usually either they arise from our senses, either auditory, visual cues or memory cues that are stored that were originally arose from our sense. The epithalamus pineal gland releases melatonin. Melatonin accumulates throughout the day, and probably at this hour, your melatonin levels are going to be high, and that feeling of tiredness is going to be in part because of the increase in melatonin, the accumulation of melatonin. The cortex contains gyra, sulci fissures, longitudinals, all of those. Here you have, this is depicting the different highways of axons that are within the brain, and these highways of axons allow different regions to communicate with each other. Here you have the visual system communicating with several portions of your brain, especially with the frontal cortex, where a lot of these executive functions, critical thinking is going to occur. It needs input from all of our senses in order for it to perform a better, a good decision. Here you have the hemispheres themselves are communicating with each other through the corpus callosum. And here you have also through the anterior commissure. And this is a view of the different lobes and with all of the specialized regions of cortex. So here's the visual cortex in the occipital lobe, sensory cortex on the parietal lobe, motor cortex in the frontal lobe, auditory cortex in the temporal lobe, and gustatory cortex within the specialized structures called the insula. The insula, as the name suggests, is it's an island. It's a small portion of cerebrum that is located not within the surface, but within the, you have to peel off this layer in order to reach the insula. But that's where the gustatory center is going to be present. And your olfactory cortex as well. All of these areas, they have a level of an association area. This association area where these inputs come together, sometimes with other inputs from other systems, and they provide richer context within it. Association areas provides context and helps you coordinate a response. There is this concept of hemispheric lateralization, meaning that there are, first of all, there are certain specializations within regions of your brain. You have your speech center within your left portion of your brain, writing, coordination of writing is going to be in the left portion if you are right-handed. Otherwise, it's going to be left-handed on the. Otherwise, it's going to be left-handed. If you're left-handed, it's going to be in the opposite end. So, the spatial visualization is going to be right hemisphere centric, and so forth. The point being is that both hemispheres, they have somewhat of overlapping function, but there is just like the fact that we are right-handed and that your brain controls everything that involves being right-handed is a hint that there is specialized areas within your brain. Basically, all of the functions that you observe rise from the structure and the neuronal specializations within your brain. If we experience functional specialization, that means that there's gotta be cerebral specialization. Cranial nerves, cranial nerves are going to be, there's going to be 12 of them, and here we have them. Let's look at this view. It's more didactic. So we mentioned that from anterior to posterior, that's how they take their names, with the olfactory being number 1, optic 2, oculomotor 3, trochlear 4, trigeminal 5, 6 abducens, 7 facial, 8 vestibulocochlear, 9 glossopharyngeal, but I hope you get the gist of it, that how they are named. Here you have olfactory nerve number 1, carrying sensory information responsible for scent. Cranial nerves 2, 3, and 4 are going to be the optic, oculomotor, and trochlear nerves. Optic nerves is going to carry the bulk of the visual information. Oculomotor is going to be, as its name suggests, in motor movement. And trochlear nerve is going to involve movement of your oblique muscles as well. Here we see them in action. Here you see optic nerve number 2, which is carrying all of that visual information. And here you observe the oculomotor and the trochlear nerve innervating these different muscles in your eye to help with movement. Cranial nerve number 5, the trigeminal nerve, is a mixed nerve. It's going to be divided into 3 different regions: an ophthalmic, maxillary, and a mandibular. And these are going to provide sensory information from your face, and it's also going to provide motor information for that help you with mastication. The abducens nerve is going to also be involved in eye movement. This is going to innervate the lateral rectus muscle. And here you have your lateral rectus muscle present here. And that's going to allow you to do this movement. Look at the middle of your finger. Facial nerve number 7 is going to be a mixed nerve. It's going to send some gustatory signals, as well as going to send, it's going to allow you to, all of the rich expressions that, all of the rich expressions is due to the complexity of our muscles, and most of them are going to be innervated by your facial muscles, by facial nerve number 7. Vestibulocochlear nerve number 8 is going to be divided into 2 branches, the vestibular and your cochlear branch. Your cochlear goes to your cochlea. Your cochlea is involved in your sense of hearing. Your vestibular branch, it goes to the vestibule, this structure that is involved in your equilibrium and motor coordination. Whenever we get to the auditory system, we'll see how this works. Glossopharyngeal is going to be involved, it's going to have, it's one of those mixed nerves and innervates the tongue and pharynx, helps you with swallowing and also nice sensory information. The vagus nerve number 10, it's involving parasympathetic function of the several of your viscera. So your lungs, liver, stomach, all of these parasympathetic functions, which involve just the regular working of your viscera, of your lungs, of your liver, stomach, those are going to involve cranial nerve number 10. 11 and 12 are the accessory and hypoglossal. Hypoglossal nerve, hypoglossal is going to be involved in tongue movement, while the accessory is going to be involved with swallowing and some innervation in the neck. Again, it's your sternocleidomastoid muscle being innervated by number 11. That's it. Does anybody have any questions?
No, I don't have any.
Oh God, it's a lot. I recommend you go over this. I recommend you go over the other PowerPoint videos. Definitely go over the anatomy textbook in order to understand all of these, and not only understand, but also just memorize all of these new terms that we threw at you. All right?
Thank you.
All right, have a good day, everyone.
You too.